Topic 7, an introduction to tissues. We've talked about atoms and we've talked about the monomers that atoms can be used to make. We've talked about the polymers that are built out of those monomers, and we've talked about structures like the plasma membrane that are built out of macromolecules. We've talked about organelles and we've talked about cells. We're going to continue up this biological hierarchy and look at tissues. Tissues are collections of cells that work together as a unit to perform a certain function. Remember that the plasma membrane is a mixture of phospholipids and proteins. Many of those proteins and even some of those lipids have carbohydrates attached to them. So those would be known as glycoproteins and glycolipids. And those carbohydrates, as you hopefully remember, identify the cell as being part of you, and also it identifies the cell as being of a particular type and belonging to a particular tissue. Unlike plant cells, fungal cells, and bacteria cells, animal cells do not have a rigid cell wall. Instead, we have a more flexible layer of proteins around the outside of the cell. And what we typically see is that we have these long filaments of protein, things like collagen and elastic fibers that we'll talk about, and they work kind of like cables or ropes, or in some cases bungee cords. And then these filaments are embedded within a firm but gel-like material. So that gel-like material would be referred to as a matrix, or sometimes it's called a ground substance. So the example you see in this picture here of these guys putting down concrete, reinforced concrete, this is similar to the structure we see in animal cells. It's not quite that firm, of course, but think of the concrete as the matrix material, and then we have these rods that run through it, and the rods are like the fiber that helps support the extracellular matrix. So when you're pouring concrete, the rods are actually resisting tension. And you might not think it, but concrete can bend a fair bit in a structure like a bridge, and we have these rods to take up those tensional forces. And then the concrete itself is going to resist compression. And this is similar to what happens in the extracellular matrix. We don't have as much rigidity, of course, to the extracellular matrix, but we have fibers that will resist tension and resist outwards pressure on the cell, for instance. So the cell will push up against the plasma membrane, and because those fibers can resist tension, they will push back. And then the gel-like substance does resist compression so that the cell can't be squished. Again, those cells are a bit squishier than something like a bridge. The extracellular matrix is highly variable. It's more variable than we see in plant cells, for instance, and other cells that have a rigid cell wall. Different cells throughout your body form different tissues that do different functions and have different requirements. And the extracellular matrix will contain differing amounts of fiber and different types of fiber depending on what that cell has to do and how flexible that tissue needs to be. But throughout the body, collagen is the most common protein fiber. It forms rope-like structures. It resists tension well, but it's also somewhat flexible. All extracellular matrices have to perform three main functions. They help cells stick together, so the proteins act like glue. They will support and protect the cell, and also they're going to connect to the integral proteins inside the cell membrane, which in turn will connect to the cytoskeleton. So the ECM is anchored to the cytoskeleton. Collagen exists as three long polypeptides twisted around each other. This is the stuff that gelatin is made of. So Jell-O, for instance, is made of collagen. Collagen is really easy to extract. You just take some cartilage and skin and hooves and all that kind of stuff and boil it up, and you extract the collagen, which of course will gel when it cools. The collagen fibers that you might see under a light microscope, if you're looking at connective tissue, for instance, are made up of tens of millions of individual collagen molecules. Those molecules are bundled together to form structures called fibrils, and then the fibrils, in this case, have been bundled together to form a fiber. In the photo at the top there, you can see a number of large collagen fibers that are overlapping in different directions to help support the cell. The fibers are a lot like a steel cable that you might find in a suspension bridge. If you take a closer look at a steel cable like that, they're made up of many, many individual wires. Here's a very high magnification image of the extracellular matrix taken with a transmission electron microscope. Notice that the collagen fibrils are running almost at right angles to each other. So they're forming sort of a grid-like mesh like the one that you saw in the image of concrete being poured. And the space in between those fibrils is going to be filled with this matrix substance, which is a gelatinous proteoglycan. The extracellular matrix is tied to the cytoskeleton, and together they're going to provide support for the plasma membrane and for the cell from the outside and from the inside. So we have proteins known as integrins, which, as the name suggests, are integral proteins. They cross the plasma membrane. They connect to actin filaments, microfilaments of the cytoskeleton on the inside of the cell, and they attach indirectly to fibers within the extracellular matrix, such as collagen. So everything is going to be tied together. Proteoglycans, remember, are mixtures of protein and sugar, but as you can see, they're mostly sugar. So we've got that chain of amino acids down the center, but then we've got lots of proteins coming off of this. These substances tend to latch onto a lot of water, and they create gel-like substances. Normally, most cells within a tissue are going to be anchored to neighboring cells, or to a basement membrane, or to connective tissues. It's not always the case, though. Blood is the most notable exception. Of course, blood cells are just surrounded by plasma. During development, we also have some cells that wander throughout the embryo, and neural crest cells are a nice example of that. We'll talk about them later when we talk about the development of the nervous system. Multicellular animals are collections of individual cells that work together. And this whole system falls apart if the cells come unglued, so to speak. So the cells need to stick to each other. What you're seeing in this colorized photograph here are some cells that line the inside of the intestine, and they're involved in absorption. And then one big cell in the middle that looks kind of strange, and it's stained kind of green, that's a goblet cell that's producing mucus. But all of these cells are attached to each other. So we can see the location of the plasma membrane there. And there's going to be elements of the extracellular matrix that are gluing those two cells to each other. Cells have to communicate with each other. This is especially important during development. So we have many different organs with many specialized tissues, and cells take their identity from the cells around them. This has to happen in order for cells to organize themselves into organs and organ systems as we develop. There are three main junctions in animal cells that either stick them together, allow them to communicate, or allow them to pass materials. The first type of junction we'll talk about is a tight junction. Now what this does is it does hold the cells together, but more importantly, it prevents material from moving between cells. So there is a gap between the plasma membrane, and it is filled by components of the extracellular matrix, but that is not waterproof. The extracellular matrix is not waterproof. This tight junction is watertight. And this is what that looks like. It almost looks like stitching. So we have two plasma membranes here. We have rows of proteins that are embedded in the membranes that are coming together and forming this watertight seal. Now let's imagine that we had some sort of fluid, some sort of substance up here that was trying to sneak through this tight junction. It can't get past these seals. We see this, for instance, in the epithelial layer that lines the inside of the intestine. And the reason it's there is to prevent materials from seeping between cells out of the intestine and directly into the blood. We don't want that to happen. We want the cells to have an opportunity to select what the body wants and reject what the body doesn't want. So this forces anything that's moving from the lumen of the intestine to the blood to pass through a cell. A cell has to choose that substance. Desmosomes are very strong attachment points between cells. So they connect one cytoskeleton of one cell to the cytoskeleton of the next cell, and they act a lot like rivets. Here we can have a closer look at one of these desmosomes. Again, they are quite rivet-like, they're very localized, and on the left you're seeing a photograph taken with a transmission electron microscope, so very, very high magnification. We've got two cells. The plasma membrane of one is roughly along here. The plasma membrane of the other one is roughly along here. Now, these desmosomes contain proteins between the gap between cells that will attach themselves through integral proteins to this massive protein that's found on the interior of the cell. So we've got all this protein here forming almost a button-like structure. And we've got that on both sides. And then these proteins on the inside are going to attach to the cytoskeleton. Specifically, they're going to attach to the intermediate filaments, that's all this dark stuff here, that help support the interior of the cell. So that looks roughly like what you see on the right if you use your imagination. Here you can see those junctions in epithelial cells from the small intestine. We've got desmosomes that are going to bolt or rivet the cells together, and then we also have tight junctions. And once again, the purpose of the tight junction is to prevent this from happening. We do not want material to seep between the cells, because once it gets down to the basement layer, it'll just go right through the connective tissue. It tends to be fairly loose connective tissue, and it will get to the capillaries and it will be absorbed into the capillaries. So we want to force any material to have to go through the cell. It has to be actively taken in, and then it has to be released from the other side of the cell before it gets to the blood. So again, the function is to make sure those cells have a chance to process what gets through. We have a desmosome, again, shown there on the right, and one more junction that we haven't talked about, we'll get to it in a moment, is a gap junction. The gap junction actually allows material to move freely between cells. We have our rivet-like attachments called desmosomes, and then we have something called hemidesmosomes. Hemi means half. These are half a desmosome. Instead of attaching to another cell, it attaches the cell to the basement membrane. We have tight junctions that form a seal between two cells. We have gap junctions that allow for communication between cells, which we'll talk about in a moment. And then we have tissue-specific junctions that hold cells together and resist separation, but they're tissue-specific, and those are called adherens junctions. Here is an adherens junction. So note that we've got this collection of proteins that forms a structure that crosses the cell membrane and attaches to the cytoskeleton. And at the other end, it has what's called an adhesive recognition site. This molecule will bind to other molecules that have the same site. And if a cell comes from the same tissue as another cell, there's a good chance it will have that same recognition site. Those cells can stick together. If the recognition sites are different, because let's say one of the cells is a heart muscle cell and one of the cells is a liver cell, they won't stick together. So we have to have the same recognition site. And this is the reason we can have isolated organs and that organs can develop at all. The stomach, as it develops, will not stick to the pancreas and commingle cells with the pancreas. They will remain separate entities because of adherence. Here's a fun little example from the world of sponges. So sponges are not quite animals, but they do have adherens junctions, and they use those adherens molecules to maintain their independence, basically, from other individuals and other species. So here we've got two different species of sponge, And what you can do is you can take a sponge and you can blend it up in a blender into a slurry of cells and then dump it into an aquarium and it will reform a sponge. It's pretty cool. If you take cells from two different species and you mix them together and then you dump them into the aquarium again, the cells will settle out. They will look for cells that have the same adherens recognition site. They will only stick to them and they'll stick back into a colony, back into a mature sponge. So pretty cool, and this is similar to what happens during our own development. As I've mentioned, cells move around and they look for their place. They look for cells surrounding them that have the same adherens molecules. All our major cell types and tissue types have their own associated adhesion proteins, and that results in this selective adhesion. Cells can only hang out with certain other cells. They're not going to attach to cells that are very different from themselves. We have one last cell junction to talk about, and that would be the gap junction. Gap junctions consist of regions with lots of very large channels that directly connect the cytosol of two cells. So you can see here we have this rosette of proteins with a big opening in the middle, and that opening is continuous right across the two plasma membranes. The cytosol can move back and forth, and anything dissolved in the cytosol can move back and forth as well. That would include ions, for instance, and ions will move across very quickly. So if this is a cell that's conducting an electrical signal, an action potential, so if it's a muscle cell or a nerve cell, the signal will move instantaneously from one cell to another. And in fact, the cells that are connected in this way behave as one cell. Things like messenger RNAs can move directly across without any trouble as well. So a messenger RNA that's produced in one cell can move into another cell and be decoded and turned into a protein there as well. Instead of communicating directly through channels, many cells will secrete a signaling molecule. And that secretion occurs into the interstitial fluid between cells. In paracrine signaling, the target is very, very close to the cell that is secreting the signal. So para means beside, these cells are right next door. In synaptic signaling, we're dealing with neurotransmitters that are moving across a synapse, a very small space between a nerve cell and another nerve cell, or a nerve cell and a muscle cell or gland, something that will be affected by the electrical signal. Finally, we've got long distance signaling, and this is what happens in the endocrine system. And the signaling molecules we're talking about now are referred to as hormones. So an endocrine cell will secrete signaling molecules into the interstitial space around it, and then they will soak into the blood. And once they're in the blood, they can travel anywhere in the body, and any cell that has a receptor for that signal will be affected. So hormones are long-distance messengers. They don't travel nearly as quickly as action potentials, electrical signals through a nerve cell, but they get everywhere. So once a hormone is in the blood, it will come into contact with pretty much every cell in the body. And if that cell has a receptor that can bind the hormone, then some sort of response will occur. Different organs and different tissues can have different responses to the same hormone. And it doesn't take much hormone to cause changes throughout the entire body. So a small amount of hormone can have a dramatic effect. Signal receptors have an area that will bind to the hormone, and they recognize the shape of the hormone. So kind of like the enzyme's active site, it'll only bind a certain substrate. Receptor molecules are quite similar. So if the hormone is the right shape and the signal receptor binds to it, generally the signal receptor will then change shape, and that brings about a change in the cell. Some receptors are embedded within the membrane and the receptive part sticks towards the outside. Others are found in the cytosol, and in this case the hormone or the signaling molecule has to cross the membrane to get to the cytosol to bind to the receptor. Here we see an example of a receptor that's embedded within the membrane. And you can see on the outside we have the site where the signal molecule is going to bind, and that's going to change the shape of the other side of the protein, the bit that sticks into the cytosol. Quite often, the cytosolic portion has an active site, and it's catalytic. It acts as an enzyme. So perhaps this binding of the signal will activate the active site, and we'll get some sort of reaction occurring that generally sets off a whole wave of interactions. So we have this small input, one binding of one signal molecule, that brings about a big change within the cell. Let's use the insulin receptor as an example of this model. On the left of this diagram, you can see that we have an insulin receptor that is inactive. It's actually in two pieces. If it binds insulin, the two pieces will change shape and they'll come together and they'll form an active site on the cytosolic side of the membrane. In fact, this molecule is going to become a kinase. That's an enzyme that will take phosphates off of one thing and stick them to another. Remember that when you stick a phosphate to something, quite often you energize that molecule. So this can energize many enzymes and activate them. So it's going to activate enzymes that are needed to break down glucose, for instance, and enzymes that are needed to do something with that broken down glucose. Also, what it does is it causes vesicles that are storing a glucose transport protein to move to the surface. And then that transport protein becomes embedded within the membrane so that glucose can be brought in. But the thing to remember is that a very small input can cause a very big change. And that's because, in this case, we have the insulin receptor activating a small number of enzymes. And then those enzymes will all activate their own enzymes, and then they'll activate enzymes. At the same time, some of the enzymes and some of the other molecules involved in these reactions might deactivate other things. So we can have a huge change in the metabolism of the cell in a very short period of time just because of this very, very small signaling input. And I'll just point out that here we've got this GLUT4 vesicle. That's the transport protein that I was talking about, GLUT4. And this vesicle is told to move to the surface so that it can embed that transport protein into the membrane and glucose can get in. Notice that with these interactions, wherever you see an arrowhead, that means that something is activating something else. But wherever you see a flat line like this, it means it's turning off something. So we can very rapidly turn on a whole set of enzymes, turn off another set of enzymes, and even do the same to genes as well. So down the bottom, you're seeing what's happening in the nucleus. And just that binding of the insulin to the receptor can shut down certain genes. Receptors aren't only found on the surface of molecules. Some receptor proteins are found within the cytosol. These are receptors that respond to lipid-soluble hormones. So those are hormones that are hydrophobic, that can cross the membrane without assistance. Insulin is not hydrophobic, it's not lipid-soluble, it has to bind to an embedded receptor within the membrane because insulin is water-soluble. An example of a lipid-soluble hormone would be testosterone. So testosterone will diffuse into a cell, and if there's an appropriate receptor there, that receptor will move into the nucleus, and then this receptor will activate or deactivate certain genes within the nucleus. So this is a receptor that directly modifies gene expression. By that I mean whether or not certain genes are used. Whereas the one we were looking at before, insulin, had its major effect on enzymes and other molecules that were within the cytosol. Now, some of them did eventually get around to having an effect on the nucleus, but for lipid-soluble hormones, usually the receptor goes straight to the nucleus and brings about a change by modifying what's going to be transcribed. Another example of a signaling system would be what happens with gated ion channels. So in the top figure here, you have a channel that is closed. It will stay closed until it binds to a signaling molecule. And signaling molecules are quite often referred to as ligands. A ligand is something that binds to something else and changes it. So we have this ligand-gated ion channel receptor that's going to open when the signaling molecule binds. So it changes the shape of the channel and then stuff can get in, ions can get in. This is very important when it comes to triggering nerves and triggering muscle responses. So it's something we will come back to and talk about in more detail. Okay, let's finally start talking about tissues, seeing as that is the topic of the day. There are 4 types of tissues that make up your body. So we have 2 that are highly specialized. One would be muscle tissue. Its function is to contract, to shorten, and that generates force for movement. Then we have nervous tissue, the most specialized of the tissues, and these are specialized cells that are conducting electrical signals or action potentials. We have epithelial tissue. Epithelial tissue is unique in that we always have a surface that's not attached to anything. So these are tissues that are exposed to the outside world or exposed to an inner space within the body. And then we have connective tissue, which, as the name suggests, connects the other 3 together. Let's take a look at each of these tissues. So again, epithelial tissue is going to be lining hollow organs and body cavities or exposed to the external environment. We have an upper surface called the apical surface, because it's the apex of the cells, that does not attach to anything. At the bottom, we have attachment to something known as the basement membrane. And the basement membrane is acellular, so it doesn't contain cells. Instead, it's basically a really thick extracellular matrix. It's made up of collagen and other fibers and other glycoproteins. Here we're seeing the classic example of an epithelium. We're looking at the lining of the small intestine. And I've mentioned most of these points of interest before, but remember that these cells have microvilli to increase their surface area for absorption. They have tight junctions to prevent materials from sneaking down between the cells and into the blood. We don't want that to happen. We want stuff to go through the cells. They've also got desmosomes, which are riveting the cells together. At the bottom of the cells, the basal region, it's the base of the cells are attached to the basement membrane. And the basement membrane is made up of 2 parts. The basal lamina, which was manufactured by the epithelium, and the reticular fibers underneath that, which was manufactured by the underlying connective tissue. And typically that connective tissue is going to have some blood vessels in it as well. There may be cells wandering around within the connective tissue. We'll talk about that later, but the basement membrane itself does not contain any cells. Connective tissue binds the other 3 types of tissue together, and also it provides support. So bone is a connective tissue, cartilage is a connective tissue. The tissues that bones use to connect to bone and cartilage, those are also connective tissues. So it comes in several different forms. We have this very tough connective tissue where bones connect to bone and where muscle connects to bone. We have some very loose connective tissue that attaches 2 tissues to each other that need to move around a lot. So areolar tissue, very, very loose, very elastic tissue, and it connects the skin to the underlying muscles, for instance, because the muscles and the skin have to move around quite a bit. And then we've got muscle tissue and nervous tissue. Muscle tissue responsible for contracting and generating force, and nervous tissue is going to initiate and transmit action potentials, which are nerve impulses. We'll talk about nervous tissue this term as well, but muscle tissue will be discussed in Biology 112. Recall that you were once a hollow ball of cells. You probably don't actually remember that, but you were, and one side got pushed in, and that gave rise to 2 tissue layers, the ectoderm on the outside and the endoderm on the inside. After that, a 3rd layer developed in the middle, the mesoderm. And these layers give rise to different tissues within the body, of course, and the fate of the cells has already been decided to some extent at this point in time. So the mesoderm is going to give rise to connective tissue, for instance. The ectoderm is going to give rise to the skin, but also to the nervous system. And the endoderm is going to give rise to the lining of the internal organs, the lining of the digestive system, and the accessory organs that attach to it. Connective tissue comes from mesoderm only. Skin and nervous tissue come from ectoderm only. And interestingly, epithelial tissues can come from any of those three germ layers. So the epidermis of the skin comes from the ectoderm. The lining of the small intestine comes from the endoderm. The serous lining of the body cavities comes from the mesoderm. Epithelial tissue plays a number of important roles in your body, and we'll spend a little bit of time looking at examples of each of the different types. All epithelial tissues tend to share a few common features. They tend to consist of closely packed cells without a whole lot of extracellular material or extracellular matrix between the cells. The cells are going to be on a basement membrane, and the surface that's in contact with the basement membrane is known as the basal surface, and it's going to connect to the basement membrane through hemidesmosomes. Again, the free surface is known as the apical surface. Epithelial tissues are avascular. A at the beginning of a word usually means without, so this is without vascularization, meaning there are no blood vessels. We don't have capillaries that go into epithelial layers, and the reason for that is that epithelial layers are exposed to body cavities and to the outside world where there's a risk of infection. So we don't want bacteria and other microbes to have direct, easy access to your blood flow. They have a good nerve supply, and quite often they're sensory receptors. So in the epithelium, we have pain receptors and temperature receptors and so on. Within your body, within the digestive system, for instance, we have receptors that can detect changes in the chemistry of the body, changes in pH. We have mechanoreceptors that can sense stretching of the stomach, etc. There tends to be rapid cell division within epithelial tissue. And if it's a tissue that consists of several layers, it's the bottom layer that's undergoing mitosis pretty much all the time. And that's because, once again, being exposed to open space, these cells get damaged, they get worn out quite quickly. The functions that are performed by epithelium are numerous: protection, filtration, lubrication, etc., etc. And we'll talk about some of those functions. The basement membrane is acellular. Again, there's no cells there. There's a lot of fibers, connective fibers. You have the basal lamina produced by the epithelial cells and the reticular lamina underneath that, and together that is the basement membrane. The function is to provide support for epithelial tissues. They have something to attach to, and the underlying connective tissue will also attach to that membrane. Also, though, the basal lamina serves as a guide during development. So during development, there are some cells that are moving around through your body, and cells that will become epithelium cells will move across the basement membrane, and it serves as kind of a pathway, a scaffold for them to find their right place in the world. There are many forms of epithelium that could be considered coverings or linings. So the epidermis is the covering on your body. The lining of the small intestine is a type of epithelium. In this case, they can be protective as well as allowing for the exchange and absorption of materials. We also have glandular epithelium, and this makes up portions of glands, the portions that are actually secreting stuff. They can be endocrine or exocrine. So exocrine means that the material is going to be secreted into an open space. So sweat, for instance, is going to be secreted onto the surface of your body, whereas endocrine means that the material is being secreted into the interstitial fluid and then it's finding its way into the bloodstream. We can classify epithelial tissues based on whether or not they have one or more layers of cells, and also we can classify them based on the shape of those cells, specifically the shape of the surface cells. So if we have just one cell layer, that would be a simple epithelium. If we have two or more, that's stratified. Strata means layer. We can also have pseudostratified. Pseudo means false, so they look like they're stratified, but they're actually not. It looks like there's more than one layer of cells, but when you look at these tissues under the microscope, there is actually one layer, but you can't follow many of the cells from the apical surface all the way down to the basement membrane. So because the cells are kind of going off in weird angles sometimes, it looks like you have more than one layer. Also, the nuclei tend not to line up, which also gives the impression of multiple layers. When it comes to the shape of the surface cells, they can be squamous, flat. They can be cuboidal, which means their height, length, and width are roughly the same, and quite often they're more rounded in appearance. They can be columnar, which means they're tall rectangles, they're column-shaped. Or we can have transitional epithelium, where we have a mix of different shapes. And this is typically in a tissue that can stretch quite a bit, and as it stretches, the cells will kind of realign themselves, and the shape of those cells may change as well. This classification scheme is pretty straightforward, and I'm hoping that by the time you've gone through this and also had a chance to look at slides in the lab, you won't have too much trouble telling me what sort of epithelium we're looking at and give me an example of where you might find epithelial layers that are simple versus stratified, squamous versus columnar, etc. Squamous cells are flat and thin. Because of that, material passes across them quite quickly and quite easily. So wherever we need to have rapid diffusion of gases or liquids, squamous cells do a very good job of that. Within the lungs, within the kidneys, within the capillaries, where we need rapid exchanges, we have only a single layer of squamous epithelium. Another thing to note about squamous cells is they resist shear forces. They resist friction. So let's say we have two organs within the abdominal pelvic cavity that are rubbing up against each other. If we had great big floppy columnar cells on the outer surface of those organs, they would be damaged. But if we have flattened squamosal cells, they're going to resist those frictional forces. So serous membranes are also squamous epithelium. Cuboidal epithelium is found in a number of ducts and glands. A really good example of this would be the ducts within the kidney. So within the kidney, we have some areas that are squamous cells where we have rapid exchange. We have other areas where we've got cuboidal cells, and we'll talk about that a lot more in Biology 112. Simple columnar epithelium is a rather common tissue within the body, and we'll find the best example of that within the small intestine. So within the small intestine, we have a single layer of epithelium that's going to absorb nutrients and water from the lumen of the intestine. And of course, it has microvilli to increase the surface area and the rate at which absorption can occur. Mixed in with these cells, we have goblet cells that secrete mucus to keep everything moving fluidly within the small intestine. We have ciliated simple columnar epithelium, which consists of cells that have cilia instead of microvilli. They're attached directly to the basement membrane, so we only have one layer. These are not as common. What the cilia do is they beat and they move stuff. So we find this in the bronchi, the smaller tubules leading into the lungs. We'll get to that in 112 as well. But these are cilia that are going to waft debris and mucus up out of the lungs into your throat so you can swallow it. The fallopian or uterine tubes are also lined by columnar epithelium that contains cilia, and the cilia are going to move the egg cell down through the uterine tubes towards the uterus. Pseudostratified epithelium appears to consist of more than one layer of cells, but it actually doesn't. So although we're dealing with columnar cells, they tend to be a little bit misshapen. They tend to be kind of spindly. So you can see that a number of these cells have rather narrow attachments to the basement membrane. You can see other cells that don't make it all the way to the apical surface of the tissue. But note that every single cell in this diagram is attached to the basement membrane. So we only have one layer of cells. You're most likely to encounter this within the upper respiratory tract, so within the trachea. And if you see pseudostratified cells, let's say under a microscope, rest assured you're probably looking at a section from the trachea, and there's going to be cilia on top. There'll be a fuzzy layer on top. It's not shown here, but there will be cilia that will move mucus and the debris it contains up into the throat so that you can swallow that debris. I'll quickly run through a few actual examples here. So here we have simple squamous epithelium, and here we're looking at a serous membrane. We're looking at the outer surface of the intestine. So that's the surface that's going to make contact with the peritoneal cavity. So we have this thin layer, we have this production of fluid that is going to help lubricate the outside of the organ. Here we've got simple cuboidal epithelium, so the cells are roughly the same size in every dimension, width, length, and height. So they're rough cubes or spheres, and you can see that we have a nucleus in each of them that is quite prominent and quite easy to see. It's right in the middle of these cells. These cells tend to be adapted for absorption and secretion, and the best example we have, probably in our slides in the lab, is from the kidney, but this is a nice sample here that's showing you part of the pancreatic duct. All the figures you're likely to see in textbooks and lab manuals that are showing simple cuboidal cells will show a transverse or cross section of a tubule. What you're seeing here is a longitudinal section. And it should be, hopefully, what you would expect to see. But it's important to get used to thinking about these structures in 3 dimensions. That will really help you when you look at laboratory slides. Non-ciliated, simple columnar epithelium is found in numerous organs within the body. It's most prevalent within the digestive system. So from the duodenum all the way down to the rectum, we have non-ciliated, simple columnar epithelium, usually with microvilli. And microvilli are those extensions, again, that increase the surface area for absorption, but also they increase the surface area over which enzymes can bind. There are a number of enzymes that are bound to the membrane of the microvilli. Also, we have goblet cells mixed in. You can't see them in this slide, but I'll show you those in the next slide. In this section of the colon, you can see lots of goblet cells. So these are the big clear looking cells. If you're seeing them under a microscope in the lab, they'll be kind of empty looking cells, or they may stain kind of bluish. But they're definitely not empty. They're filled with lots of vesicles that are packed with mucus, and that mucus is going to be dumped into the lumen of the colon by exocytosis. We need a lot of mucus in the colon, and that's because, of course, the contents of the colon are being formed into feces. The last bits of water are being drawn out, so we have a solid substance now, and it needs some lubrication in order to continue its trip towards the anus. What you're seeing here is ciliated simple columnar epithelium, and it can be rather difficult to distinguish this from non-ciliated simple columnar epithelium that possesses microvilli. So the cilia and the microvilli are both very thin extensions of the cell. They do entirely different things though. So the cilia are going to move back and forth. They're going to move material. Under very high magnification, you may be able to note that the cilia are thinner, but maybe the best way to figure out whether you're dealing with cilia or with microvilli is taking note of where the sample came from. So we will see this type of epithelium in the lower part of the respiratory system. In the bronchioles, we see pseudostratified ciliated epithelium in the trachea, and we will see this sort of epithelium within the uterine tubes. I mentioned before that the cilia are going to move the egg cell, and perhaps even a developing embryo, down the uterine tubes or fallopian tubes towards the uterus. And here's the pseudostratified ciliated columnar epithelium. So we're seeing it from the trachea. Again, it looks like multiple layers, but it's only one layer because all of those cells, even if we can't trace the entire cell, all of those cells attach directly to the basement membrane. Here's another nice image of pseudostratified ciliated epithelium. You can see that we've got cilia at the top and we've got these large goblet cells. And the goblet cells are connected to the surface and they will secrete mucus up onto the surface. And what the mucus is going to do is it's going to form a layer on top of the cilia, and then debris will get stuck to that mucus. This mucus is very sticky. So it could be bacteria cells that you've breathed in, it could be dust that you've breathed in, and that debris is going to be moved in one direction up to the throat by the beating of the cilia. So the cilia will all move in a coordinated fashion to move this stuff upwards into the throat. Now let's talk about epithelia that are actually stratified. So we've got more than one layer of cells, and we're going to classify these epithelia by looking at the shape of the top layer, the apical layer. So with stratified squamous epithelium, the top layer will consist of squamous cells, so flat cells, but the deeper layers don't have to be squamous. In fact, they're usually cuboidal. The cells that are at the bottom that are actually attached to the basement membrane are the cells that are going to be dividing by mitosis, and as these cells divide, they push cells upwards, they move up towards the apical surface, and they flatten out as they do that. We find this in areas where we have a lot of frictional forces quite often. So within the esophagus and vagina, for instance, there are frictional forces that can be countered by having a thick layer, stratified layer of squamous epithelium. Some squamous epithelia can become keratinized, and what that means is they become filled up with keratin. So the cells towards the top fill with this really tough protein that is resistant to frictional forces, helps keep bacteria out, and provides some waterproofing. So this keratinized stratified squamous epithelium is generally referred to as skin, and we're going to spend a whole topic talking about that. But realize that there are some areas that are partially keratinized. So for instance, within the mouth, the surface of the tongue is partially keratinized. And that makes sense because, of course, your skin is going to be continuous with the inner surface of your mouth and other openings on your body. Stratified cuboidal epithelium is rather rare. There's not too many examples of it, but we see it in large sweat glands known as apocrine glands. We see it in mammary glands, which are actually modified sweat glands. And we see this in the ducts that lead from the salivary glands into the buccal cavity. Stratified columnar epithelium, also pretty rare. Again, the top layer is going to be columnar, but the layers lower down won't necessarily be. And we find this in the male urethra, and also we find this in the ducts of some glands. Here we're seeing stratified cuboidal epithelium in an apocrine gland. And here we're seeing stratified columnar epithelium. In this example, it's lining the interior of a very large duct, but it can be found in other places as well. Other examples would include the conjunctiva, parts of the pharynx, the anus, and also the uterus. Notice in the drawing, once again, only the upper cells look columnar. The other cells all look cuboidal. Transitional epithelium is stretchy. It permits distension or enlargement of an organ. So we find it lining the urinary bladder, for instance, because the urinary bladder does expand significantly when it fills with urine. We find it lining portions of the uterus, the ureters, and also the urethra. Stratified squamous epithelium can be quite thick. So the epidermis of the skin can consist of over 40 layers of cells. And the top cells are going to be heavily keratinized, which means they fill up with this tough protein called keratin. It's the same stuff that makes up your hair and fingernails, and the cells are also dead as well. Other areas of the body that have stratified squamous epithelium won't have any keratin in them. So that's the case, for instance, in the vagina. It's the case for the clitoris and also the glands of the penis, the head of the penis. Now, if the penis is circumcised, then the glands does actually dry out and it does develop keratin. It becomes keratinized. We have other parts of the body where we have partial keratinization. So for instance, the tongue and the lips have some keratin, but they're not fully keratinized. In transitional epithelium, we've got a jumble of cells of different shapes. The cells will stretch most along their long axis, and the long axis of these cells runs in different directions. So that allows this tissue to be stretched in many directions. Glands, both exocrine and endocrine, are modified epithelium. So epithelium that's been modified for secretion. We can have epithelial layers like the lining of the small intestine, for instance, that contain single cells that are glands. So that would be goblet cells, and they produce mucus. But we can also have very, very large glands like the parotid gland that produces saliva. And it's made up of millions of modified epithelial cells. Generally, these cells are cuboidal or columnar in shape. We can classify glands using 4 criteria. First, we could look at the number of cells that make up the gland. And then we could look at whether or not the gland secretes into a duct or just dumps its product into the surrounding interstitial fluid. We can look at the shape of the gland, and finally we can look at the mode of secretion. So how do they get this material out into the world? Goblet cells are single-celled glands. So they do everything a gland would do. They're just one cell big. They are quite larger than the cells that surround them in most cases. So what you're seeing here is a villus from the small intestine, and you can see these large cells that are releasing a whole lot of material, mucus, through exocytosis. Exocrine glands secrete material onto a surface. So that could be the exterior surface of the body, if we're talking about sweat, for instance, or earwax. Or it could be an internal cavity, if we're talking about saliva and digestive enzymes. They do this through ducts. So they have tubes that carry the material to the surface. Exo means outside. Think of exterior, or the exoskeleton that surrounds an insect. Endocrine glands secrete materials into interstitial fluid surrounding the cells, and then that material is going to seep into the bloodstream and travel throughout the interior of the body. And endo means inside. For the next 2 levels of classification, we're looking just at exocrine glands. Now we can have unicellular glands like goblet cells, but for the most part when we're talking about glands, we're talking about multicellular structures. And these structures can be fairly complex. We can have branched glands that are quite large that have several lobes to them, as we'll see. We can talk about their cross-section as well. Are they tubular, or are they shaped kind of like little sacs? Are they alveolar? To use structure as a classification scheme, what we do is we look at one of these ducts and we look at the shape or structure of the tissue that attaches to it. So in these diagrams here, you've got the epithelial layer at the top there, and also the duct shown in purple. And these are regions, of course, that are not secreting. So the secretion is happening from the pink tissue. We can have 2 different shapes for the tissue that is secreting. We can have tubular, or we can have acinar, which is a sac-like structure. And I have to admit, the 2 look quite similar in this diagram. We can have simple tubular. We just have a tube that's connected to a duct. That can be coiled up into simple coiled tubular, and you'll see that in sweat glands when we look at those in the skin. And we can have simple branched tubular. So we have branches coming off of that duct. We can also have simple branched acinar. Now we can also have branches on top of branches. So what you're seeing here is we've got additional levels of complexity because we have branches coming off of those simple branches. This is what you'd see in something like the pancreas, we've got a huge mass of these little acini, these little sacs that are producing enzymes, for instance, and they are all connected by a whole tree of ducts that lead back to one large duct that is going to empty the contents into the intestine. Finally, we can classify exocrine glands based on how they discharge material, how they go about actually secreting stuff. The most common exocrine glands are the merocrine glands, and these glands release material by exocytosis. So vesicles fill up with this material, they fuse with the plasma membrane, and the stuff gets dumped outside of the cell. Apocrine glands accumulate material in the apical part of the cell. So material moves up to the top of the cell, the part of the cell that is exposed to a free surface. And then what's going to happen is that top portion is going to pinch off, and then it's going to break down and release material. In holocrine cells, what's happening is the entire cell, after it's filled up with material, is going to pop, basically. It's going to die, and it's going to discharge all of that stuff. Let's look at this in action. So at the top here, we have a merocrine cell. And again, this is how most of the cells in your body work. What happens is we have vesicles coming off the rough ER, and they're going to contain proteins. That's going to be moved to the Golgi, where the proteins are modified more, and then the vesicles that come off the Golgi are going to carry that material to the surface. The vesicles will fuse with the plasma membrane at the apex of the cell, and that'll dump the contents. In apocrine cells, we have those vesicles accumulating at the apex of the cell again, but the vesicles don't fuse with the plasma membrane. Instead, we get this asymmetric division of the cytoplasm, and that's going to cause that top portion to pinch off and break down and release its contents. Interestingly, it seems that humans probably don't have any of these cells. We've identified them in other animals, and it was originally thought that humans had them. In the early microscope work, people thought they saw this occurring, but in more recent studies, it's been shown that no, it's probably exocytosis that's occurring. Now, because of that, we have some cells that are referred to as apocrine, even though they probably aren't. We have apocrine sweat glands, which are found in the armpits and in the groin, and they produce a more viscous, thick sweat that bacteria like to dine on, and that creates the smells associated with those areas. The mammary glands were thought to be apocrine as well, and they're still referred to as apocrine glands, even though they're probably not. Finally, we have holocrine glands. These are used to produce oil, so oil that may find its way onto the surface of a hair or onto the surface of the skin. What's happening here is we have cells at the apex, the apical surface of the epithelial layer that are going to die and dump all of their contents. So you can see the top cell there is making a lot of material. It's filling up with these vesicles full of secretory material. And then the cell is going to undergo apoptosis, programmed cell death, and the entire cell will break down and rupture. And then notice below the cell that's in the process of dying, we're going to have new mitosis occurring, and new cells will move up and replace that lost cell. So here you can see that happening in 3 different acini. The first one, merocrine, and the example they're using there is the pancreas. The second one, apocrine, and the example they're using is the mammary gland, although this is most likely wrong. It takes a long time for textbooks to catch up with recent research. And then we have a holocrine gland, and what we're looking at here is the sebaceous glands that dump oil onto the shaft of a hair. And finally, here is our terminology list.